Okay. Welcome. I'm looking forward to this course. I have a bit of a cold. I tested negative, so which might change at some point. But yeah, just so you know, you're safe. So yeah, hi, I'm Thomas. Welcome to Brain Imaging. In this course, we'll try to teach you how to look inside someone's head, not by topping their heads up or peering deeply into their eyes, but by means of using a scanner. What I like about this course is that I get to teach the stuff that I do every day. So what this allows me to do is also in this course really show you what we in the lab are doing on a day-to-day basis. And that to me really helps me sort of bring the course and the material alive. So I hope that at the end of the course you will agree. At any point during lectures, if you have any questions, just raise your hand and I will try to answer your questions as best as I can. I've started putting up things on Canvas. A few points. So Canvas has a syllabus in there. It doesn't have a bunch of modules or anything like that. You can go to Perusall for reading of literature. Right? So we do that online through this Perusall interface. We are not necessarily grading any of your reading, but we are going to notice who participates in the reading more than others, and so that will be like a bonus sort of thing at the end of the course. And you can follow the Zoom links for the lectures by just clicking there. I just discovered that, so I'm just saying it as a novelty. So let me start by just introing what we're going to do during the course. I noticed that in the PDF I put online, this still had 2021 in there. Don't really know how that got there, but it's 2022 already. Yeah, so brain imaging. Also, for those people joining online, if you ask a question in the chat, I will try to notice that as best I can and get back to you. Okay, brain imaging. Who of you have ever been inside an MRI scanner? 2, 3, 4. Excellent. I hope that this was for research and not clinical purposes. But maybe those of you have already seen the insides of their own heads, which is a pretty magical thing to be able to do, I always think. So what we'll try to do in this course is to show you what it takes to be able to make these images of the inside of someone's head, and then also what we need to do to make sense of what the brain is doing. And so brain imaging, we're not just wanting to look inside people's heads in terms of the tissue, like a doctor would, for example, like a radiologist would. We are interested even more so in what the brain is doing. So in this course, we'll be focusing on functional brain imaging. So this thing isn't working. So I'll just try to fix it. Yay, it's working. I hope that all problems that we encounter in the course will be so easily fixed. So, well, I'm Thomas. If you have any questions, raise your hand during the lectures. And if you have any questions outside of the lectures, drop me an email on this Gmail address. I'm monitoring that day and night for any messages that you might have for me. It's an actual like specific teaching address, so I'll know that it's you as a student. And if you put this brain imaging thing in the subject line, you can be sure that I'll know which course this is about. Yeah, just to, you know, for the intimate introduction, this is my brain. It's the outside of the gray matter of my brain, to be precise, and you'll learn during the course exactly how I generated this image, right? So that's the type of thing that we'll be doing, taking these images and trying to, taking these raw images from the brain and trying to make beautiful figures like this. Even do like functional mapping of the brain. So here's an image of my own personal retinotopic maps. This sort of hierarchy of visual regions that we have in the brain. They're mapped, like these are maps of visual space in the brain, and you can actually map this out in an individual brain. So these are mine. Nice to meet you. So just as a further introduction, right? So this is my most recent publication, and as you see, I'm really involved in human brain imaging, and we're trying to use a bunch of methodologies that I think are used more and more in the field. So my hope is, or my intent is, and what I expect is that if I take you guys by the hand and show you what we're doing in the lab, this will actually be a very nice introduction for you to learn about what the state of the art is in the field that I'm going to teach. So at some points, it may feel like I'm sort of playing the tune of my own research a bit, but that's just because I think that really exemplifies what the field is doing. And also, of course, that's what I'm able to explain best, because that's what I'm doing every day. Now, in this course, we'll have two TAs playing roles. Both of these are PhD students here at the VU. They're called Jet Lagerman and Ron van der Klundert. They are both PhD students here doing like behavioral and imaging research themselves. So they're going to TA the practicals and the reading of materials. And I'll get into like what exactly the course content is. So our topics of research are fMRI. So we do a lot of scanning, fMRI scanning ourselves. Especially Ron is doing a lot of scanning. And our interest is mainly in the visual system. So also in the course, there will be a bit of a skew towards, explaining how the brain makes us see. I don't consider that a bad thing, not just because it's my field and I wouldn't, right? But also because a large part of the brain is visual, right? So it gives us a really beautiful sort of test garden to really explain how the brain works. We understand vision really well as a general sort of sensory modality, and we understand the brain's visual system also really well. So it provides us with a really nice way of showing you how to use the fMRI scanner, the MRI scanner. So I just want to highlight also that we are a research lab, and we collaborate a lot. Actually, my position is both at the Spinoza Center for Neuroimaging, which is like a Royal Society Institute just outside of the city, near the AMC hospital. So that's where the scanners are. And we collaborate with people there on like how to do fMRI, and our fMRI experiments are conducted there. And we're also very much interested in like how the scanner works. And we'll get into that in a later lecture. But there's a lot of physics and engineering behind making this huge magnet create these images for us. And so we'll teach you a bit about that. And that's what we collaborate with here in the city. Of course, we are here at the Vrije Universiteit. So we work here in the cognitive psychology department. Our offices are on the other side of the building here on the same floor. And we try to blend our fMRI experiments with detailed behavioral experiments, right? The function of the brain is to make us behave. So relating those two as directly as possible is what we aim to do. We're also very much interested in modeling what the brain is doing, like having some mechanistic understanding of what the brain is doing, and we collaborate with a lot of people at AI, especially at the UvA. So a lot of like deep learning people we collaborate with, and we try to understand the brain from that perspective also. And so as many of you are AI students, if I'm not mistaken, that is something that we'll see throughout the course also. So I'm hoping that that will be an area of interest for you specifically also. We're also collaborating with a few institutes in the US, mainly on like techniques and this AI sort of thing. I'm just putting this up here because, well, just to show you that as a lab, you sort of connect with people all throughout the world. People do similar things, and you're inside in this sort of network of researchers. And similarly, if you, okay, I'll just stay here. Thanks. That was late. Okay, but for the rest, I'll stay here. Also, many of you need to do or will do internships or thesis research parts of your in your studies. And so, if you want to go abroad, we have an international network to, put you somewhere if you want. Okay. I hope this sounds better. Okay. Now, when I was studying, I always just wanted to know, like, okay, what am I supposed to do? Right. That's all nice. You as a researcher being all geeky about this, but I just want to, get my diploma at some point. So I want to clearly outline what we're expecting from you and what you can expect from us in terms of course materials, exam materials, grading, and so forth. And so forth. All right, so just an outline of the course. Excellent. Briefly, and then we'll I'll try to explain a bit about the course in terms of the content more. So I am totally fed up with this whole pandemic thing, and I'm expecting you are too. You've borne the brunt of this whole thing, I do believe. So as students, I hope that we can sort of get away from this whole lockdown thing. I at least, and the people that will be giving guest lectures, will try to do that as much as possible here in person. Also, the practicals that we'll have, we'll try to do those as much in person as, in hybrid fashion. And I hope that that will sort of allow us to take steps towards normalcy a bit more. Okay. Now, specifically, we have tried to sort of set up the course such that we can do both, right? So you don't have to miss anything if you have to stay at home, if you have to go abroad. You can follow these lectures over Zoom. I am recording them all, so they will be on YouTube also. That's for the lectures. For the practicals, I'll explain in more detail, but they are also, you can do them at home. You can do them on your laptop here. You can do everything on your own pace. And where it's necessary or where it would add something to your learning, we can have like face-to-face discussions here. We can also have discussions on Canvas. Wrong course, maybe. I don't know. And at the end, what I was hoping we could have is, so yeah, I'll get into this in a bit, but we want to have this sort of collaborative project, right? So you can work together with a few of your peers to do a project. We have a couple of those lined up where you put into practice the things that you've learned during the practicals of the course, but there's a bit of an interaction thing going too. That's our goal. Yeah.
So the project is different from the practicals.
Yes.
And
Yeah.
practicals of analysis is something.
Yes. Yeah. I'll explain in more detail in a bit. Yeah, but a good point. It's always, when you're asking a good question, you know that it's a good question if the person does this click, and then the answer to your question is on the slide, more or less. So not precisely, but good question is all I'm saying. Anyway, so what will we be teaching you, right? There's always a balance in a course. You can make a course really theoretical, right? In which case we try to impart you with knowledge, right? And knowledge is, university, it's a very lofty goal. But in many cases, especially when it comes to research, the actual meat of what you're doing is not just knowledge, it's know-how, right? And that is a much more practical type of thing. So we try to sort of give you both. Now, in terms of knowledge, we want to really give you a strong background in that okay, an fMRI experiment, what does it consist of? For example, what are the data, right? What you can and cannot do with fMRI data, and consequently also what you can and what you cannot learn from an fMRI experiment, and the different factors that this depends on. Yeah? So this includes things like designing experiments. There's a whole zoo of different types of experiments that you can do in a scanner. And so we hope that we'll give you a bit of ideas about the types of things that you can do. Yeah? So, some exam question that I might think up is like, let's say you want to research this and this and this. This is your research question. How would you do this in terms of an experiment, right? And then you would have an idea of what you can and cannot do in an fMRI scanner. And so you could, come up with an experiment that you might do. That was imaginary. I'm not going to explicitly do that. Oh, yeah. So, Abdullah, so there's indeed 3 practicals on the roster and 4 practicals like in practice. I'll get into that in a bit. Okay, so that was the knowledge part, right? So, and then how do you interpret fMRI? What does it mean when people show you these pretty brain maps? What is that, right? And that's all knowledge, right? Along with like, what have people done in the past and that sort of thing, reading papers. But what I think is maybe even more important is this know-how part, which is how do you, for example, work with fMRI data? Like I give you a data set, what can you do with it? We're going to show you how people are used to analyzing fMRI data. There are a bunch of standard analyses that people perform, and those are like the backbone of many more complicated analyses too. So we'll try to give you a background in these standard analysis ways. Because once you have those nailed, you can much more easily understand many of the more advanced sorts of analysis also. So what we'll do is we'll try to give you this know-how really in terms of practical experience. Okay. So what does that mean? In terms of the structure of the course, what we're doing is we're having 2 lectures per week, once on Wednesday and once on Friday. I will record these, right, and I'll put them online on YouTube. And the idea is for this entire course, and I'll get back to that in a bit, I think, is that we will have more or less 2 parts to this course. In the beginning, we'll try to give you a lot of background
We'll try to teach you these basics, like the standard analyses that people do. And then at the end of the course, we can be more sort of broad scope. And we'll start reading specific types of literature, looking at more advanced types of analyses, that sort of thing. Yeah. That also is a separation in terms of practicals, right? We're going to give you these sort of relatively well-defined practicals in the first half of the course, so that you learn these standard analyses. And then we're going to move to that project in the latter half of the course, where it's more up to you, and you can make of it what you want. Yeah? So we're just going to give you a bunch of papers, for example, show you an outline of a project, and then say, you're on your own. So that's really the sort of evolution that we're trying to go for during this course. Now, what's the material in the lectures? Well, so like I said, the first half of the course is really like the basics. I'll explain the basics of what we're doing with the Fmri scanner and so forth and so forth. And then later on, the lectures will be more like guest lectures. So I have a bunch of people that I've already recruited to give guest lectures on specific topics, namely their own research, so that you get an idea of these more advanced topics from the people that are performing the research themselves. We're also going to go into discussions of very specific parts of the literature in the end of the course, like really to show you like these are the future directions of where the research field is going. Now, in total, there are 3 practicals scheduled. So this is for you, Abdullah. So in the roster, there are 3 practicals on some Wednesdays from next week, and then every second week. That's here, I believe, also in the mornings, so not like here in the afternoon. And so these 3 practicals are just opportunities for us to meet in person, right? So you can say, okay, I'm just going to work on these practicals on these specific Wednesday mornings. That's when I'm going to come here. I'm going to open my laptop, do the practicals, try to finish them in that time, and then go home and party or whatever. But you can also say, I don't know, these things are simple. I'll just do that on my own, and you don't have to be there at those practicals. Yeah. So what this means is that there isn't also a necessary link between the practical sort of documents that we have and these mornings. So there are 3 Wednesday morning practical sessions in the roster, and there are going to be 4 practical notebooks that you go through. But you can go through those in any order if you like. That's all open to you. But these practicals in the roster are just our opportunities to, sit next to you, wearing a mask or not, I don't know, and then, explain to you what we're asking of you in these notebooks. Yeah. So what I would do myself is just, try these practicals out on my own. And then, if I have any specific questions or I want to dive a bit deeper, then I would come to the Wednesday mornings. But feel free to do whatever. Yeah. Yet Ron and I will be here and online during those sessions at least. Yeah. Oh, no location is assigned. Huh. Okay, I'll make sure that we have a room. Thanks, Daria. Yeah, and so all throughout the course, what we're doing is, what I've done is I've uploaded PDFs in a Perusal library. You can link to that in the Canvas page, right? You can just go through and link to it. If they're okay. And there's about 2 or 3 PDFs of papers that we'll read every week. And again, Ron and I will be monitoring your Perusal reading. So if you have any questions, you can ask them in the text. And we'll try to answer them, those questions or those remarks that you have there. But to me, that's always been a very, it can be a bit annoying reading from a screen. At the same time, it's very nice to see people like really having interactions through Perusal. Yeah. For the first couple of weeks, yes. So there are 3 PDFs I've already uploaded most of the PDFs, so you can look in the library of the course on Perusall, and you'll see the PDFs that we are going to read also later on in the course. Yeah, but for the first few weeks, they are already signed with their respective deadlines. Yeah. I have to say, like I said, right, and like it's stated in the syllabus also, important, grade alert. The grading is going to be mostly the exam at the end. I'll get into that in a bit, and the projects that you'll do at the end, right? So those are the things that we will be basing our grading on. The Perusall, not so much. So there being a deadline for reading assignments, you know, whatever, right? We are pacing the reading such that if you have a specific reading assignment, that will most likely align also with some specific notebook that you would be doing in some practical, for example, right? So it makes sense to sort of maintain those deadlines, but they're more of a target for you than anything else. Yeah.
Because the detailed timeline is very common, right?
Yes, very good point. I'm still working on that. So last year, people found some of the YouTube clips really annoying, and you know, on second thought, I did have to agree. So I'm sort of pruning the YouTube watch list a bit, and I wasn't finished with that. But I'll try to finish that this week, so it'll be clear to you what YouTube clips to watch at what point.
Yeah, but is reading what more Perusall.
Yeah. So yeah, that's a bit shitty. That's separate. So Perusall has this internally, but you have to like go to Perusall, and only then do you see the deadlines. But it's not like they're automatically linked back to Canvas to some module structure or anything like that. So yeah, be aware of that. Those are separate sort of entities. Yeah. But you have a good point. I think I'm going to try to make the effort to like highlight the assignments. It's just that when you change it somewhere, like if I were to change an assignment deadline here, then I'd have to go back, and I tend to usually forget those types of things because I'm a researcher. Yeah. Okay, so did I just do something wrong? Oh yeah, so these notebooks and many things like that, also some practice data, stuff like that are on this GitHub repo that, you know, you can feel free to fork or clone or whatever. Yeah, not too important. So that's what we will be offering, right, in terms of the smorgasbord of different educational possibilities during the course. But what are we going to ask of you, right? That's always important. If I were you, I think that were important. So at the end of the course, there'll be an exam. And this exam, it looks like right now, but, you know, this is the view, and they're sort of, you know, weird with exams and whether these are in person or proctored. There were problems before. So I'm trying to get to the bottom of this and get reassurances that the exam at the end of the course will be proctored on TestVision. So you can also make it from your home. That is something that I definitely prefer to have. And in that case, so I'm hoping that will go through. In that case, we'll have a combination of multiple choice and open questions. Yeah, so the bulk of the exam materials we will quiz in sort of multiple choice fashion, right? But then you know, where it's about things like the exam question that I just made up of, like, think of an experiment where you do this and this and this, and this is your goal. That type of question is really cool if you can just, you know, write a little story, a couple of sentences on how you would approach this. So to me, that always gives me a much better idea of whether you've really grasped what the course is about. Yeah, so that's why we're mixing these multiple choice and open questions in the exam. So now, what's the materials of the exam? Well, the backbone is these lectures. Those are supplemented with the content of the practicals, right? So that's knowledge, know-how thing, right? Then we're going to have articles discussed and read on Perusall. Those are also part of the exam materials. I won't go into, so sometimes these things are going to be relatively detailed. I'm not going to quiz you on stuff like equations, like on page 6 of article blah. what was this equation, variable, this, whatever. I'm not going to do that sort of thing. Right? But you know, they are going to, these articles are going to exemplify specific parts of the exam material that I find important, and so they are part of the exam material. They are, you know, open to quizzing. And then finally, these lectures of mine will be supplemented with these YouTube modules by this in this YouTube channel that I'm still, you know, selecting the exact episodes of, and those will also be part of the exam materials. Yeah. So this these YouTube modules of this Wager and Lundquist thing, it's a pretty good course. They go through the basics relatively well. Jeez, what I just do? Yeah, so they go through the basics relatively well. And that's why I thought it would be great for that to supplement what I'm teaching you here. It's always good if you have multiple people telling you more or less the same type of stuff, right? Because you just get two different viewpoints, and it makes it easier for the material to stick, is my impression. It is a bit outdated though. So these YouTube videos were recorded, I think, in 2015. And as you will learn, the field has moved on quite strongly. There are very fast developments happening in the field. And so, yeah, I'll try to highlight whenever they're saying something that's already outdated. Yeah. Okay, so then this research project in the second half of the course, right, where you can really test your mettle, if you will. The idea is to have small teams. I'm saying 3 here. It can be 2, it can be 4, whatever you like. I don't care much. Where we have created this sort of Jupyter notebook where there are 3 main chapters representing 3 different projects, and you choose a project and then run with it as a team, right? So you just analyze the data, and we've all set it up such that will be, you know, that'll be easily doable. It runs in Google Collaboratory, so it's not on your laptop. If you have like a shitty laptop, it doesn't matter because Google's running the data analysis. Like the actual compute cycles, like you have to program it yourself. And so that will give you a way to put into practice all of these things that you've learned during these practicals, right, in the first half of the course. And then what you end up doing is you write this report in that Jupyter notebook. And so you can explain what you've done in these text blocks. You'll get an idea of how that works if you don't know already, you know, as you go through the practicals. So that's that. And then so I've set this up such that in the end you'll have this rubric that you can aim for while you're writing, and you can also like sort of review reports from your colleagues, right? So the idea then is that you do one of the 3 projects, right, and then you review 2 others, the other 2 projects, such that you actually get exposed to all 3 projects' contents, either by doing it yourself or by sort of grading other students' work. That's the idea. It might be the case that at some point we run out of time and then this can drop off. Yeah, I'm not like, I don't want to be a slave driver and say like, you have to like grade each other's, you know, that's what we're being paid for too. Whoa, what is that? I have, oh, sorry, I have an enormous amount of questions all of a sudden. That was closer to the laptop. Yeah, so there will be a Zoom meeting for the practicals. Sorry, this is annoying. Yes, there will also be an overview of the articles that's mostly the deadlines on Perusal. Yes, YouTube videos, there will be a schedule for that. Oh yeah, repeat the questions. That's stupid. Sorry. Yeah, that was all about yeah, the practicals. And change the exam date. Yeah. I'll check, but that's gonna be a hard one. Yeah, so I've heard that the exam is immediately after some other exam or something like that, right? So yeah, so I'm hoping that the exam can be online. And yeah, I don't know what EDA is, but I hate them already. Yeah, yeah. So there's 30. That's quite. So there's 30 minutes between the EDA exam. I don't know. Many of you will know what the EDA is. Yeah.
Yeah, it's a proportion for AI students.
Oh yeah.
Yeah, every AI student has to do that because this is an elective, so people are asking whether we can change the time of this exam.
Okay, I'll start contacting the admin people that know more about this sort of thing. Like I'm a researcher. I don't like these things are usually. It requires a certain amount of teaching for you to really understand the inner workings of teaching at the university. I don't think I teach enough for that. Or yeah, so I'm usually a bit like what? But yeah, there's that. Please not earlier. Okay, yeah, I'll try to. Yeah, that makes sense. Okay, so yeah, I hope that. Okay, I'll make a note of that exam. For now, I propose that we have a brief break. After that break, I will, tell you about what's in the course. Right? Great. Yeah. So let's be back like in 5 or 10 minutes or so. Yeah. Yeah, I've restarted the recording. So here we go. So those of you that have been inside an MRI scanner before, was that for a cognitive science experiment? Yeah? Who's been in a fMRI experiment? You can raise your hand. Who's been in an fMRI experiment as a participant, not as a, I don't know what. Yeah? So what did it feel like?
I did it as a test person because I'm working on the project, and it's a bit weird. Like you're laying in also all the participants are always saying that they're a bit anxious before, and then you have to stay in it for a long time. So yeah, it's not the most comfortable thing.
Yes, I agree. You also have been? Yeah. Can you echo this, or do you have any other experiences you want to?
Well, I was with you. It's like a bit weird because it's quite small.
Okay. Oh, that's cool. So you have your own brain. You've seen inside your own brain. That's pretty awesome. Yeah, this is what people say. It's tiny. It's uncomfortable. It's hard. I don't know if you've ever tried, but to remain really still for 5 minutes is insanely hard. And so if someone asks you to be still for an hour or an hour and a half, that's almost impossible. But you should try. Like you should not like an hour and a half. Just just for fun. Like you're sitting, you're at home. You try to be still. Like lie down. Try to be completely still for 5 minutes. Like not move your head. Your head's the most important thing, right? Because that's what we're scanning. But like if you try to do that, you'll notice that over time you get this itch. Like you need to ooh, sort of feeling is what you get. It's really weird. Yeah, yeah, definitely. Yeah. So we have a bunch of documentaries that we play for people on a screen, like in the scanner. So an anatomical scan is just, be still, but you don't have to do anything because, we're just making a still image and it takes 10 minutes or so and then it's fine. But yeah, so we have a bunch of, the one that I like best is Monkey Kingdom. I've seen like many documentaries in the scanner. There's I used to work in another lab where they were so stupid to play Wallace. Do you know Wallace and Gromit, the movie? No? It's this hilarious sort of slapstick stop motion thing. So you don't want to play that in the scanner while people are, supposed to be still because they will laugh. And that's like the worst motion of a head that you can have for imaging. So yeah, you can actually see this, like the images get really blurry and completely. Anyways, this was a total segue. Yeah, it's not a natural situation to be in an fMRI scan. I think that's the takeaway from this brief discussion. And this is something that we, as researchers, forget, right? How much of an awkward and unnatural situation we are putting people in. People just say, oh, we're doing this, blah, blah, and then they, forget. We scan people's brains while, something. And so whenever you're interpreting results from an fMRI experiment, realize that this is the case. Like people are in this unnatural situation. And that's in some sense, you can't get around that because you're asking people to do an experiment. But it's important because it limits what we can do, right? We cannot have people walk around with like multiple ton scanner on their heads, right? It's just, you cannot do that type of research. It means that, we're very much limited to only those types of cognition that you can perform while lying on your back with your head immobilized, right? And I don't know, that's not that much, right? There's a lot of things that we cannot do because of this. I just want to, basic point. Maybe I should have waited with that point, but there it is. Cool. So we can look inside our own heads, right? If we get the images, which I don't think is very is necessarily ethically okay, right? If you send your subjects, your participants, their images of their own brains, you would not be allowed to do so if they turned out to have like a brain tumor, for example, right? Then that image gets sent to physicians like a radiologist, and you cannot show people their own brains if that's the case. So if you don't get your brain scan in that case, that there's something wrong. So I think that's not something you're allowed to do as a researcher, yeah, ethically. But so save that image. Yeah. Well, yeah, you can, but there's a protocol. Okay, this is totally outside of this lecture, but we're here. So you scan someone's head, right? And you notice that this person has something weird, right?
I'm sorry, can you repeat the question?
Oh, yes, sorry. The question is, why can't you give someone their own brain's image when there's like a hole in their brain somewhere or something like that? Let's say you see a brain tumor. There's a strict protocol that we should follow if this happens. So you save the image, it gets sent to a radiologist who diagnoses this scan. So they know exactly how to do this, and they're much better at it than us researchers. And 90% of the cases, like 99% of the cases, the radiologist will just go like, yeah, this person had a cold, so there's something near their frontal lobe, it's not a problem. So something like that will happen. But there are these cases where, there is something wrong and you should be seen by a doctor, which is why when you participate in an MRI experiment, you always have to list your GP, your general physician, your huisarts, because if they do find something, the radiologist will contact your GP who will then, arrange a meeting with you, and then you will go into like a clinical, thing. They will start treating you for what they found. You'll go into extra testing and that sort of thing. Yeah, but that would be part of that clinical sort of trajectory. Yeah. And so people are really sort of anal about this. So if you were to give people their image, whenever, right? So whenever they participate, and you'd have to withhold their image if it was still being seen by a radiologist, because otherwise they might be able to look at it themselves, see something weird, and freak completely out, right? And so you don't want that. And so this practice of giving people their brain scans is very normal, and it is becoming less and less normal just because of this. And I think this is a general tendency in the field. People are becoming much more aware of these types of ethical considerations in general, right? Also in terms of, male-female balance in the participant population, age range in the participant population. Many of these things people are becoming attuned to that more and more. So that's the reason. Yeah, is that clear? Sorry for the segue. Yeah. Anyway, so apart from these ethical considerations, which are boring and beside the point for now, fMRI is awesome because you can not only look inside someone's brain, which as a still image can be very beautiful, but still, it doesn't move. Well, yeah, so it moves, but okay, this is going wrong. So it's a beautiful, anatomical structure. But what's really cool is that the MRI scanner can be used to image function, right? The actual stuff your brain is doing. And that is like almost magical, right? We can now, with the MRI scanner, introspect in a way that doesn't involve us closing our eyes and trying to, what do I think I'm feeling sort of way, but actually see what's happening inside our own brains. And that is truly, truly awesome. So this was invented, I'll get into that now or later, I don't know. It was invented like 30 years ago, and it's sort of a part of this evolution of brain imaging that's been going on for almost 100 years. So fMRI is just one. Oh, geez. Okay, I'll get into these questions after this part of the lecture, okay? Because I'm just taking way too much time already. I'll get back. Okay, so in around 1990, so somewhere between the 1990s and the 2000s, people discovered that we can use this MRI scanner that we already used to create images of inside people's heads to really look at brain function, right? To see which brain areas are active and which are not. And that was a huge thing, not just because of its novelty in sort of a first principles way, but mostly also because these MRI scanners were already in every hospital, right? They were already being used by clinicians to diagnose things like brain tumors, to look at things like multiple sclerosis, Alzheimer's disease, where you take anatomical images of people's brains, and they are very important for things like diagnosis and research. They were already in all of these hospitals, like liver imaging, knee imaging, MRI was already a thing, anatomical. And so what that meant is that now all of a sudden all these cognitive neuroscientists could like take their experiments to the hospital and start scanning people. And this made it huge, right? So it's expensive to do, but it's also everywhere. Right? So people could just go to their hospital and run an experiment. And that really created this enormous flux of fMRI studies over the last 20, 30 years. It really has been growing tremendously. And if you look at popular science literature, what this has meant is that we are all pretty much used to seeing pictures of stuff inside someone's brain, right? So like the pleasure center, like the love region of the brain, that sort of thing has really become a sort of pop science. Everybody's used to seeing that type of image. And what is done is it's placed for everybody their minds inside their heads, right? And this is not a trivial thing, right? So where do we place our minds? It's been like a philosophical thing down in the ages. Like people at some point thought that the brain was, convoluted like that, so it could exude heat more efficiently, right? So placing our psyches, our minds in our heads, this firmly, you can actually credit MRI with that. But if we think about things like the love region of the brain, that is hopelessly simplistic and wrong, right? And if you show people blobs in brains, they tend to be like, oh, wow, that's really convincing. But when, the story is total and utter bullshit, right? So we don't want that. What we're going to teach you is to sort of pierce through that sort of magical ring to it and really give you an idea of what you can and what you cannot do with brain imaging, with functional brain imaging. And what I think this, and so you could sort of say like, okay, yeah, what does all of this mean, right? If it's just what we sometimes call blobology, right? So you do an experiment and you find a blob in the brain, you go, this is there. And it doesn't tell you anything about what this is functionally, right? It doesn't tell you how this works. It just tells you it's in some place in the brain. But I think some undercurrent in all of these papers really is very valuable in that it is showing us how our brains function in a very fundamental way. Yeah? I don't know what you concur to here. So, in principle, a course called brain imaging, even a course called functional brain imaging, could do many different things, and we are focused on fMRI. So why are we focused on fMRI, and what other things are there? And I could give you an entire lecture, like a couple of weeks of lectures on everything that we can do to measure the brain, right? That could be construed as brain imaging. I won't. I'll just give you a general overview of the things that we can do. And this is a beautiful figure used in all of the textbooks, and many of you will have seen it before. What you see here, I'm just going to sit closer to the laptop so that they can hear me. What you see here is a space-time diagram, not space-time in the sort of Einsteinian sense, but you have time on this axis and you have space on this axis. They're both scaled logarithmically because that's what you do. Time here goes from milliseconds or tenths of milliseconds to like lifetimes over here. Yeah. Then in terms of space, what you do is you go from like this, what's this? Millimeters. So this is 1 millimeter, micron. Let's see, where is the micron? Micron and smaller, like nanometers somewhere over here, right? I think, yeah. And so what this means is that if I want to use a measurement modality that has a temporal precision of years, but I do want to know stuff about entire brain regions, I would have to find the research area that does brain lesions, right? When a whole lobe of the brain gets destroyed by some lesion, by some bleeding, for example, it will take years. So that's what you'd be interested in. Yeah. Well, that's the field that you would have to choose. Now, on the other hand, if you are very interested in something that happens really, really fast, and you want to measure the entire brain, you have to move to the left here, right? So you want to go to a shorter time scale. And so you end up doing something like EEG and MEG, where your temporal precision is close to a millisecond level, and you have whole brain coverage, but you still don't really have spatial precision. Yeah. So in the end, what we would want is to be all the way over here, in which case we would know everything about, and then, with whole brain coverage, you would know everything that happens in the entire brain very fast. There is no such measurement device. right? So usually when you go to fast measurements and very local measurements, what you do is you sacrifice this idea of whole brain coverage. Yeah. So for example, single units, they're actually measuring single neurons, a single neuron at a time. So that leaves out, let's say, 150 billion other neurons that you're not recording. And the same goes for patch clamping, which is like intracellular. You're like looking inside the cell even. Now, yeah, so that you have all of these different things. We could go into all of them. That's not really important. What we are interested in is fMRI, right? Oh yeah, so explanation, sorry, temporal resolution and spatial resolution, right? Why has fMRI taken such a flight other than it being there in all of these hospitals? It's actually because it is non-invasive and because it provides a really, really compelling mixture between these two dimensions of spatial resolution and temporal resolution. Okay. Now, so you see fMRI imaging here. Okay. What does this mean? It means that we can do something on the order of seconds, right? See, something like that. You could put someone in the scanner for days if you feel like it. That would be really uncomfortable, wouldn't it? It's been known to happen, but you can do things on the entire brain, like on the whole brain, the scale of the whole brain, and you can zoom into things like the layers of the cortex, or like really, really tiny parts of the brain if you push things. And the really nice thing about fMRI is that you can actually cover the entire brain, like measure the entire human brain. You can do so at around a second time resolution, and you can do so at a spatial resolution that is around millimeters, right? So we are usually scanning somewhere here. You notice that we're trying to get there. Like, this would be awesome. Too much data, but it would be awesome. We're trying to move here, and we are usually scanning around here in this corner. And if we sacrifice this whole brain coverage idea, then we can go smaller. So we can go there. Maybe we can.
I'm sorry, we cannot. Well, I don't think I can see your.
I'm in the way. I'm sorry. Yeah.
So here and here doesn't really make sense.
Thing that I expanded. I'm sorry. Yeah. Here. Do you see that?
No.
Okay.
I can see the laser pointer by the video.
Yeah, no, you're right. Yeah, so the left bottom of that purplish thing that I expanded with the arrows, that's where you want to be, because there you have the highest temporal resolution and the highest spatial resolution. Yep. And what's really cool about this is nothing else gives you this. Yeah, just to contrast MRI or fMRI with these other modalities, EEG and MEG give you a lot of time resolution, but you have 64 electrodes, maybe 128 electrodes. MEG, you get 300 like squids, like measurement points. What that means is that your spatial precision, especially inside the brain, isn't that good. You get like 0.5 centimeter, maybe 4 millimeters. And that is blocky. That is really blocky. It's just not the level where you would hope you would be. So you get a bit of time, but still you don't know where things are. Yeah?
Just to be clear, the upper left corner is the most flexible.
Lower, like left bottom here.
No,
this.
like from the whole.
Oh yeah, you would love to have like. So this is what people can do in you know, the fruit fly. The larvae of the fruit fly, you can genetically engineer to express light to, you know, you can do like whole brain microscopy at a cellular level at something like 200 hertz. So you can actually see all of the neurons firing in sequence, each and every one of them. So that happens, but you can't do it in a human because we're too big and bulky. And we can't genetically engineer people. There's a bunch of, you know, practical considerations, possibly even ethical considerations. But yeah, so you know, animal studies, they can go here. But yeah, so this means more data, right? So you get more samples per space and more samples per time. You want to go to the left bottom because it's more. more information. Yeah. And so this is a diagram that is, I think, really informative because it tells you where the strengths of things are in measurement modalities and where their weaknesses are. So just to, there's also PET imaging. It can be very specific in a biological sense. So people have used it for functional imaging. So I'm just going to contrast. I just contrasted EEG to fMRI. Now I'm contrasting PET to fMRI. That used to be really good in the 90s. People used a lot of PET imaging. The problem with PET is that you're having to inject a radioactive substance into your subject, which means that you cannot do it every day because you'll, get like Chernobyl type things happening. So that's not allowed. And the thing with the fMRI is that you can just keep putting people in the scanner over and over. They will get nauseous, they will get annoyed, they will get mad at you, but there's no long-term effects. It's not like you get magnetized, like you are when you get vaccinated, for example. Yes? Yes, and so as we get better, and I'll get into what better means, we're trying to push the envelope, moving towards that direction, yeah, because more information, and more information is better. Yeah, good. Now, when you do EEG, for example, you're measuring, the E stands for electrical encephalography. So you're measuring actually the electrical signals from the cells that are firing, right? And that gives EEG its fast properties. But in fMRI, we don't measure anything electrical. We measure things by means of magnetism, but in a later lecture, a colleague of mine will show you that this has nothing to do with the electromagnetism of the brain itself. The signals that we measure with fMRI are metabolic. And what that means is that we measure not the electrical signals of the brain, we measure the blood flow in the brain. Yeah? So the measures that we take are BOLD, standing for blood oxygenation level dependent signals. Okay? Yeah. So the idea is that when a neuron activates, it will consume some oxygen, and it will tell the cells around it, like, hey, I need some extra oxygen. And so the tissue responds, and that causes the bloodstream to respond. So the vasculature has this change due to the neural activity. And then that changes things like blood flow and oxygenation. And it's an incredibly complex cascade of things, meaning that we are measuring something metabolic. And this is also very important in terms of interpretation, right? I was saying that I will teach you what you can and what you cannot do with fMRI. What you cannot do is perform measurements of neural activity. So if you read the literature, people will say this. They will say, we measured neural activity in humans performing X and X task. They're wrong. You can only measure metabolism at specific locations in the brain using fMRI. Okay? So this is important. We're not measuring the neural activity itself, but some proxy of it. Yeah?
I have a question. Isn't there a tiny bit of a delay?
Oh, yes. Yes.
Or
That's
how does that matter then? If you say that if some neurons want some oxygen,
yeah,
there is some blood flow, there should be some delay, right?
Yeah, there is. I'm not going to answer this question now. I'm going to go to the next slide, probably. Which meaning, again, it's a very good question. Right, so yeah, exactly. So when you, and so I'll get into this a bit later in a separate lecture, but so this cascade and this indirectness, meaning that we're looking at a derivative or a proxy of the neural information processing that we're interested in, this goes pretty far. Right, so it means that this activation to some neural activation, so the signal that we measure in response to some neural activation is really slow. It takes over 20 seconds to unfold. So yes, there is a delay. and not just the delay, this whole cascade of stuff that happens as a result of this neural activation not only is delaying everything, it's also smoothing it in time. Yeah? So what that means is time is not our friend in fMRI. You are always going to be limited in the time domain because you will not be able to resolve things that are closer together in time for less than about a second. And even then, you need to be really aware of what you're doing. Yeah? So just to go through this, right? So you get a neural response to some input. You get oxygen demand, which changes. This goes through glial cells, and there's this whole sort of biological cascade of different sort of transmitter systems like nitrous oxide and that sort of thing. So what that means is that the vasculature in the tissue will change in terms of its blood flow. It will also change in terms of its oxygen content. And what you can then do is sort of, okay, you can model this. It's called a balloon model. Not important. We won't go into that in detail. So you get like a change in the volume of blood, and you get a change in the ratio of hemoglobin oxygenation in the blood, and together you get what we call this bold response. Yeah. So that's a lot of words saying it's indirect, and we measure for an event lasting about a half a second or so. We measure this type of time course. Right. This is slow AF, right? This is a almost 5 second delay to the peak of your response from the event that caused this response. Okay, so anything really fast in terms of experimentation, you really can't do with fMRI. Yeah, you need EG or you need, whatever. But yeah.
You said it takes five seconds to see the effects. So what
Yeah, so that's a good point. This shape, we call this a bold response shape or a hemodynamic response function for HRF. So all of these things I'll say more about later on. So I'm saying 20 seconds here because this thing has a stereotypical shape. It goes up and then it goes down and it stays down for a while. And then it's more or less back at baseline around 20, some people say even 30 or 40 seconds. Okay. So, and this is just a response to a single event, right? We'll talk later in later lectures about what happens when you have another event here. Oops, right? Because then you have the gap of this response in the peak of this, and it gets all muddled, right? And so to combat that sort of iffiness of having a slow, bold response, that's where most of the analysis paradigms come in. Yeah? So, but we'll teach you all that, so don't worry, okay? So this is what that response looks like. And again, underlining this idea, we are not looking at neural activity, right? We are looking at this, at the blood flow. I think here it is. I just actually accidentally clicked this out. But so this is what happens when someone dies and you pour like plastic into their arteries, right? So you infuse the vasculature of their brains with plastic. Then you let the plastic solidify, and then you throw the head into sulfuric acid, so that eats away the entire head. And this is a vasculature cast of the inside of someone's brain. This is what we're basing our measurements on, right? And of course, this plastic cannot really like go into the tiniest of blood vessels, but there's like this beautiful web of tiny vessels inside all of the gray matter of our brains, where the neurons are that use the energy. Right, so just to show you, this isn't, it's not neural activity, again, that we are recording, but these vessels are tiny and they're everywhere. So there is a relatively good correspondence between where the neurons are firing and where we see this sluggish bold response. Yeah. Great. So balloon model is just, it's just a word, there's no actual balloons. Yeah, sorry. So, do I have time? I still have some time, right? 10 minutes, 15, something like that. 15. Oh, great. Thanks. Yeah. So the MRI scanner. Some of you have been in it, others have not, in which case, feel free to contact the Spinoza Center. You can actually volunteer for experiments if you feel like, what was it, uncomfortable? Yeah, so those, if you want those things. Okay, so every hospital has at least one. It's a very standard clinical device. And so what we do is we just repurpose it to perform cognitive science experiments. Now, the idea of using a big magnet and a bunch of radio receiving coils, like antennas, to create images that you can't see because you would have to cut things open to actually see them, right? This nuclear magnetic resonance imaging that received the Nobel Prize in 2003. It is truly magic what we can do with a scanner. It is like insane. I'm getting goosebumps. But it's like the amount of understanding that goes into making a scanner and making it work is really, really awesome. Like as a species of animal that was, not able to do like read or write 5,000 years ago, to be able to make a machine like this really is a testament to our understanding of how the universe works. And this all sounds incredibly grandiose and hyperbolic, but it's not. If you think about how you need to understand magnetism, how you need to understand electricity, and how you need to be able to manipulate them in detail inside living tissue, that is just, awe-inspiring, the things that you can do. And so to me, it's always like on a par to these particle detector things like the Atlas at CERN, right? These huge machines where there's so much engineering going into it that it's completely insane. The scanners are like that. Sorry, that was another segue. But what can you do with a scanner? And we'll have an MR physics lecture at some point by a colleague of mine that actually does that thing, and you'll get an idea of this. And I'm still talking about the awesomeness. So you'll get an idea of how complicated things are, and it's always easy as a non-physicist. So I studied a bit of physics, but I'm not like an MR physicist. For someone that doesn't sort of speak that language, it's always easy to go like, yeah, sure, it's complicated. But if you try to get the idea of how much goes into it and how much engineering has gone into it, you'll get a sense of awe that I'm hoping that you'll get from that lecture too. Anyway, let's say you have a scanner and you can use it to do fMRI. Turns out that someone discovered this, there's a hospital next door, and you can start doing this experiment in living humans. It's pretty cool. But the scanner is used to create many, many different types of images. You can do it of dead brains, but there's nothing against doing it in living brains. So especially this idea of anatomical images, you can make many different types of anatomical images with one and the same scanner. And that's one of those really cool things about this. It's not fixed to a single type of image. This scanner is able to do many different types of things. It can do things like functional imaging, it can do anatomical imaging, it can do flow imaging, it can do many different types of things. It's a very versatile machine. And what happens is you have this sort of fixed machine and you just run different programs on it, different software on it. It tells the different magnet systems, different coil systems to transmit and receive things differently, and thereby producing completely different images. Not even images sometimes, you can do chemical spectral imaging. So it's just a very, very versatile machine, and everything depends on the programs that you let the scanner run. It's like, you have a printer scanner thing attached to your computer, right? You can print out anything as long as you just plug it into the USB. You can even do it wirelessly nowadays. I'm getting old. Anyway, so the thing is, different programs called sequences make the scanner do different things. You can create different types of images, and so forth, and so forth. And the precision of the images depends on the strength of the magnet. It's a bit silly, but it really is the case that the stronger the magnet, the better the scanner. Yeah? But it's also similar to, you know, a difference between a standard sort of Volkswagen Golf and a Formula One car, right? So here you'd have your Volkswagen Golf, and here would be your Formula One car, right? It's really hard to do grocery shopping in a very large scanner. Okay, that's taking the metaphor a bit too far, but so you can, so there's a lot of things that have to go just right for the image to pop out as beautifully as it could in a perfect situation in an 11 Tesla scanner, right? You have to have a whole team of engineers like you do for a Formula One car, right? You have to have an entire team of engineers, and they have to really monitor all the systems, what they're doing, and then you can get a beautiful image from an 11 Tesla scanner. Yeah? On the other hand, if you have this, which is in every hospital, it's very easy plug and play to get reasonable images quickly. So there's this trade-off there, but the things that you can do with scanners such as these are pretty cool. And I'll try to show you some examples during the course. Clear? Yeah, I know. Like, if any questions? Really, yeah. So I've never been in an 11 Tesla scanner. It's one of my bucket list items, I guess. But I've been in a 7T a lot. There's a big difference between these. Like in a 7T, you go in and you're in, like only your toes wiggle on the outside and the rest of you is inside the scanner. Whereas this is just a little ring, you know, you're only in the scanner this far. Whoop. This far. Oh, I lost the battery. I'll click on the trackside from now on. Okay. So we run this program, right? We can run any program on the scanner, but the program that we run for fMRI, for functional MRI, gives us images. Yeah, that's what we ask the scanner to do. So we don't just get images like you do with like a camera that are 2D images, right? What we get are 3D images. And so you in essence, get a cube of values for every scan that you take. What this means is you have this 3D box in X, Y, and Z. And the elements of these images are not called pixels for picture elements, but they're called voxels for volume elements. Right? It's a very simple sort of way of thinking about it. And here's some examples of the different types of scans that you can perform using this scanner, right? So different programs that you run on the same scanner can give you different outcomes in terms of the image. Now, in this case, these images were created to be exactly the same in terms of spatial dimensions, but they use a different contrast, as we call it. I don't think it's, yeah, you can sort of see it on the top, right? Again, this is me. And I'm going to try, because ethically it's easier for me to use me as a, you know. And so we have these three directions in the scan, right? We have the sagittal scan, easy to remember. Sagittal seems like an awkward word, right? But I don't know who of you has the zodiac sign of Sagittarius, right? So that's the archer, right? I forget which month it is. I'm not an astrological person, but so the Sagittarius stands like this, like he pulls the bow in this direction, right? So you have to imagine that the bow line just cuts through his face in the middle. You get an image like this, right? So that's the sagittal view, the sagittarial view, if you will. So if you were to wear a crown, right, you would see that crown best as a sort of 2 points on the top of someone's head here, right? So this is the coronal viewpoint of that 3D box, right? So we have 3 dimensions, of course, then we have another one, and this one is axial because that's the main axis of your body from top to bottom, yeah? So axial, coronal, and sagittal views. I was talking about these different contrasts. What you see, This is called a T1 weighted image, and we'll get into that in the physics lecture later on. T1 stands for something, believe me, T2 does also. And what you see is that they look completely different, right? They are taken minutes apart of the same brain. So this is the same object that's being imaged. But here you can actually see my nose, right? Or you can see my spinal cord, or all of those things you can see. Whereas here, that more or less drops out, and you don't really see the outside of the head. Right? And so what this means is you can actually visualize different tissue properties with the same scanner in the same format, right? By just running a different program. And we'll get into why you need these different things. But for example, if you want to diagnose someone's brain tumor, you would run this because it pops out better. And that has to do with the fact that something has more water or fat or something like that, right? So these basic biological tissue properties, they determine how things look in these different sequences, in these different scans. And these are just two examples. There are many, many more. So if you go to a hospital and you get your brain scanned, I hope you won't, because I hope, you would get the scan of the entire brain at a resolution of 1 millimeter. That's the standard, right? And we're constantly pushing this. The transverse plane is that kind of depends. That's hard. I'll talk about that in a bit. So as we're pushing the techniques, and that's really happening, we have this sort of constant evolution of the engineering behind this. We are pushing this type of anatomy to higher and higher resolutions. Our standard at the 70 is whole brain at 0.6. That means you get much crisper view of all of the anatomical structures, and I'll go into that in later lectures, really show you what the differences are if you increase the resolution. And if you want to do very, very specific anatomies of specific parts of the brain, you can go to a resolution of 0.3, 0.2 millimeters. That is tiny. It's actually so tiny that usually a heartbeat will move the brain more than that, right? So your heart is actually pulsating blood into the bottom of your brain on every heartbeat. All of the brain more or less moves about this much all the time on every heartbeat. Right, so we're at the limit of what you can actually image, more or less. Yeah.
Well, if it's a living Tesla, then why is it still useful? If it pulsates more than.
Oh yeah, so that's a good point. Like at some point you'll hit a limit, right? Like you might say, this is not usable anymore, right? But then someone smart will come along. That's really the story of MRI in general, in terms of its history, is always like, shit, we can't do this, right? And then it takes a couple of years and people are getting used to the status quo that this you can, this you cannot do. Then someone smart comes along and says, hey, but what if you do this with this coil and I got this thing where you can, and then, hey, right, all of a sudden we can. So let's say a scan about this heartbeat thing, right? I just made this up. So let's say you can scan for all the, continuously, and then of course you get blurring because the brain moves all the time, right? What you could also do is say, I'm going to measure someone's heartbeat, right? And as it beats, I'm going to always take a snapshot of what I want to record 100 milliseconds after the heart pulsates, after the heart beats, right? That means I'm going to catch the brain always in the same, period of that, phase of that motion, right? And that do away with blurring if I'm really smart about it, right? So yeah, you're right. That is a really good point. And this happens all the time. People much smarter than at least me, probably not you guys, are making up new stuff all the time. And it's pretty amazing. Yeah. So also, I just use this clicker thing, but I think I broke it. So it's important to realize also, we're looking at volumes. Important conceptual points. Looking at volumes means that if you start out with a 1 millimeter resolution, that means 1 millimeter in X, 1 millimeter in Y, 1 millimeter in X, 1 millimeter in Y, 1 millimeter in Z. right? And that means you have 1 cubic millimeter, right? If you go to 0.3 millimeters, right, it's about the amount of tissue in that voxel, right, in that cube. If you go to 0.3, you have like 1/3, 1/3, 1/3. You have to multiply these, right? So the amount of volume inside of the 0.3 millimeter cube is only 1/27th of what was in that original 1 millimeter voxel, right? And that volume is where your tissues are in, and that volume is also where your signals originate. So we're actually really pushing the boundary. Right? It doesn't feel like much, this difference of 3-fold, but it's actually a 27-fold difference. People forget this. Okay, so what does fMRI data look like? This is what it looks like. It's blocky because if you want to have a very high resolution image, it takes more time. And if we're interested in function, we want to sample fast. So we don't have much time for every image that we take. So that means that we tend to think, okay, a relatively blocky image is just fine, we'll just do it. Because we can actually acquire that image from the brain on every second or so. Right? And then we can measure time courses. So what I'm showing you here on the left is a brain that I'm going to be clicking through, and on the right, you will see time courses. And so I'm going to click it, and it'll play a movie, I hope, and then you'll see the types of signals in time that we have for each of those points in the brain. Right? So this is important to realize. When we do fMRI, we have 4D data. We have 3-dimensional images at every time point in the scan, in the data. So 4-dimensional data. I'm just clicking around. Hey, there's signal. There's no signal. There's signal. Bunch of signal. Signal. See? You have noise, lots of noise, and then there's some signal. Yeah? And this is something that you'll see during the practicals, and we'll go back to during the lectures also. There is this trade-off here. We are measuring a combination of signal and noise in these voxels. And the goal of most of our analyses is to find which is signal and which is noise. Yeah? That's the general idea of all of our analyses. Yeah?
Could I see in general what was noise and what was
No,
signal?
no, no. I'm just, oh, yeah, no, I'm just.
Said, oh, there's this and there's that.
Yeah, no, you're right. Yeah, no, this is, so you have to know the experiment to know what is signal, right? Because you have to know what is likely to have generated the brain activations. I'm saying signal here very confidently because this is an experiment where there's something on the screen. You can guess how many times. 8 times, right? So there was a signal generated 8 times in this experiment, which I knew and you didn't. Sorry, I should have said. But so you see that this voxel here has very strong activations to this stimulus that we showed 8 times during this experiment. And you'll actually learn to analyze this specific experiment. I think actually one of the practicals has exactly this data in it. So this is a visual experiment, and the voxels that I'm clicking there in the back of the brain, which contains the visual system. So this is, yeah. But these other voxels that we were looking at, for example, like here-ish. Oh, that's not right. There were some crappy voxels somewhere. So this is noise, right? We expect this type of thing. If you measure only sort of crap like this, you know that that's noise. And well, I won't go into this, but there are many different sources of noise also. Some of those sources of noise we can actually sort of, work out of our data. Others we cannot. But this ability to say this is noise and this is signal is really the heart of most of our analysis strategies. Yeah? And of course, this sounds a bit, weird, and I didn't even tell you that what the signal was, but we'll get there. Now, this is just another example of how we can use the scanner. This is actually DTI for diffusion tensor imaging, where at every voxel, we don't measure a time course, but we measure the directions in which water is likely to diffuse inside that voxel. And that allows us to, you aim this for the white matter. It's intended to analyze the white matter. And you can actually trace the fiber connections throughout the brain using this type of scan. Same scanner, different software program that you run, different sequence. Right, so you really, so what you see here is the diffusion of the water molecule sort of thing. But you can actually trace exactly, for example, things like connectivity between different regions, that sort of thing. Right. What you can also do is actually image speed. So this is flow imaging of the arteries around, I think this is, not really sure. I'm going to say this is around the brainstem. So you have the carotid artery going up and then linking through around the brainstem. And you can actually, for every location inside those arteries, you can tell using the MRI machine how fast and in which direction the blood is flowing there. So if someone has like a blockade somewhere, this is used in the hospitals to find those. Okay? This is just to underline this machine can do whatever, right? It can also, if you aim it for a specific location, you can find the concentration of specific neurotransmitters at that location, like a chemical measurement. So it is really, really versatile. So in this course, I'm going to try to speed things up a bit. So, in this course, we'll teach you how to analyze these types of fMRI measurements. And it's important to realize that it's more than just a single step. You really need to know the experimental design, like I forgot to tell you just now, right? You need to know the experimental design, and then you can say what is signal and what is noise. And you have to always keep in mind what your goal is, right? What am I trying to find? Because it's so much data, you're very likely to get lost if you don't keep track of your goal. And there's many, many ways of going wrong. So you're wanting to conclude something somewhere at the far end of a pipeline of processing steps. It's a chain of events, a chain of processing steps. And if you make a mistake somewhere early on, you get the principle of, for the subsequent stages, garbage in, garbage out, right? So you have these entire pipelines of processing. We'll get into those, and we'll show you a bit about what can go wrong and why we need to spend a lot of time optimizing this. That's because, I don't know if you've heard, the psychologists among you will have heard, in this neuroimaging field and in general, in sort of medical research, science in general, there's this reproducibility crisis or replication crisis. So this hits neuroimaging really hard because it is so complex, right? Because you have this enormous pipeline of different processing steps. What that means is you can have all these different processing steps and look at whether the end result confirms your hypothesis, right? So you can then rerun the entire thing, just changing a little bit, right? And see whether it now confirms your hypothesis. And you can keep doing that over and over until it actually tells you what you want it to tell you. And then you publish, right? You go to Nature and say, we found this. Well, you could not have found that if that was your course of action. But people did this a lot, right? So this tinkering is really problematic. We'll go into this in a later lecture. Also, these enormous amounts of processing, if you're sloppy, you can just, by accident, be analyzing activations from outside the brain if you didn't do your stuff correctly, for example, right? So we're really, there's a movement going on how to make all of this more reproducible. And we'll teach you the best ways of doing that, right? So our goal is, and what's happened before is that after you've finished this course, you do an internship in an imaging lab somewhere, and you will be able to teach them how to do it, right? That's the goal, right? And this is not because, you know, this course is so awesome. It's just that, you know, many labs started 10 years ago and they have their own little pipeline of doing things. And nowadays there is this sort of open source software development type thing going on where people are joining forces, and these things are much better than what people can make on their own. So we'll really introduce you to these concepts and how to use these types of things. So this is like how this processing pipeline that we helped implement works. Just, you know, we'll get into that later on. And like I said, like this, okay, I have half a minute. Like I said, the YouTube videos are a bit old, and there are new developments. I just want to highlight one of them, because the state of the art is being pushed further and further. So if you use a newer scanner, you can do more. And one of the things that people have started doing is to do laminar resolution MRI. I'll show you more about that in later things. You can do, you know, people have started doing very large open science projects. So you can actually download enormous amounts of data for free and analyze them in your own way. If you have an idea, you can do this. We're specifically working with the Human Connectome project in the lab, which is 1,200 subjects, each scanned for over 5 hours, both at 3 Tesla and at 7 Tesla. We're also working with this natural scenes data set where 8 subjects were scanned for 40 hours, not in a row, but, you know, 40 sessions together. There's like even larger things there. And data analysis techniques are also improving, you know, very, very quickly. And the thing there is that people used to do relatively sophisticated things with data analysis, but it would be like this, you know, sort of closed off sort of thing. Whereas nowadays, everybody knows Python, everybody knows, you know, general GitHub and programming practices. So people are starting to share things more, and things become more collaborative and open source projects. And people are doing their best to make sure that all of these things run in a reproducible way. Okay, that was boring, but we'll get to that later. So this is an example of how small you can get your voxels to be. This is at 0.7 millimeters. So you get really increased resolution. That's where we're going. And we can really start to compare our fMRI results with animal electrophysiology because of this increased precision. So here's an example of me running through a functional image. Right? So this is acquired every couple of seconds, and you really see things like the layers of the different brain structures. That's where we really are. And we're doing a lot in terms of also trying to model what the brain is doing computationally. Right? So this link between AI and neuroscience for us is really tight. And I'm not going to have time to show it now, but I need you all to go to this website and see how this type of computational approach to brain imaging is powerful. And then, yeah, we can also, what we're really starting to look at is also not just how every unit in the brain works, but also how they as a whole, as a network, work to give rise to things like cognitive processes. Yeah, so that's like one of the ways in which people show these connectivity results. Okay, so you don't need to go now this instant, but there's already a couple of papers that have assignment dates on Coronal. You can start reading those. The first ones are a bit backgroundish, and we'll get into more detail as we continue. You can start watching the YouTube videos, and I will put a detailed list of which clips to look at for which lecture also on the syllabus this week. Yeah. Thanks. Sorry for running overtime, and I'll start answering questions. You can go. Hi, I'm gonna answer all of these questions in an announcement. Okay, I'm sorry. This is yeah, it took too long.
So how could you monitor the reading? Like, is it just you have to read the PDFs?
Yeah, so I can show you. Wait,
Well, I saw them already. I went on Carousel.
Oh, yeah, you did.
Yeah,
Okay.
but I just
Then-
wanted to know if it was like we just have to read them.
Yeah, you can. So what the website does is it has this AI that's between you and the PDF. So it will notice when you select things and when you comment on them. So it will sort of count your interaction with the PDF, and it will also monitor how fast you scroll through the thing. So yeah, based on that, it will sort of say you've been reading well or you haven't. That sort of thing. Yeah, it's not that, I don't think it's that complicated.
How many PDFs we will have?
I'm aiming for like 10 in total. So I'm thinking like 2 per week, something like that. But let me know if it's too much. I mean, yeah.